A clinical trial testing a novel gene therapy for a rare neurological disease has been put on hold after one of the participants in a Phase 3 trial developed a bone marrow disorder that can lead to cancer. The pause, announced Monday by the trial's sponsor, Bluebird Bio, and mandated by the U.S. Food and Drug Administration, FDA, was taken out of an abundance of caution, the company's president of rare genetic diseases, Andrew Aubenshine, said in a recent quarterly call. The mutation causes fat to build up in the brain, where it breaks down the insulating myelin that allows neurons to communicate with one another. Because the gene is on the X chromosome, women typically have at least one good copy, so the disease primarily strikes men. Left untreated, it causes damage to hearing, vision, cognition, and coordination. It is often fatal. Bluebird's gene therapy uses an engineered lentivirus to correct the mutation associated with the disease. While other virus-based platforms using retroviruses had previously been linked to cancer among patients, it is only recently that a lentivirus has been implicated in such an outcome. In February of this year, Bluebird Bio paused another trial, one for a blood disorder, after 2 patients developed. Most in the field were hoping that we would not see such an event with lentiviral vectors. Harry Malich, a gene therapy researcher at the National Institutes of Health, tells Science, adding, "I don't think anybody's been." After a round of chemotherapy to reduce the person's bone marrow cells, the treated cells are infused back into the patient. Thereafter, the patient's stem cells produce healthy blood cells with a functional copy of the gene for ALDP. The therapy entered the market in Europe last month following a previous safety and efficacy trial that included 32 patients. A second trial, the one that has now been paused, was set to finish in 2024. Speaking on the call, Bluebird Bio's chief scientific officer Philip Gregory said that one patient in the second trial developed myelodysplastic syndrome, MDS, a blood disorder that sometimes leads to leukemia, and another 2 had abnormal bone marrow cells that could progress to MDS. When scientists examined their cells, they found lentiviral DNA inserted at a site in the genome that has previously been linked to MDS in retrovirus-based therapies, suggesting that the virus may have caused the changes. To ensure the gene produces enough ALDP in the brain to be an effective treatment, the researchers needed a strong promoter, but as a consequence, the promoter had off-target effects, turning on other genes in the area around the mutation, including cancer genes, Gregory speculated. He adds that this particular incident shouldn't preclude scientists from pursuing other lentivirus treatments, as the issue seems to come down to design, and Cohn doesn't know of any other lentivirus therapies that use the same type of promoter. This is a severe adverse event, she says, but adds, we should never lose sight of the fact that so many patients have been helped. Despite this rare incident, the findings could help scientists and researchers design safer and better vectors for the future. Please support my channel to grow by pressing subscribe button and the bell icon. We will notify you technological news. Thank you.